Hello everyone. In this session, I'll be going over a few other important topics for the upcoming FMGE exams. Right, myself, Dr. Varsha Gupta. So I'll be taking this session for the biochemistry. Few days back, actually, I have already taken this session. You can Google it on the YouTube also. You can search it over there, right, about the previous PYQs for the FMGE, and I highly recommend you and request you to go through all those questions again, because recall questions are very, very important for your examination, because questions are not going to be same, but they tell you the high yield topics. So like I humbly request you to go through that video again. I'm not taking those topics again, but few more topics, so that we are covering a larger aspect of the biochemistry. Okay, so I went over like so many previous year questions, and what I have figured it out, every year they are asking maybe like 2 questions from the vitamins section itself. So these are all the questions I could get, right? So I thought it is better that we start this session from the vitamins topic itself, though I'm not going to go over all the vitamins, but a few ones, and then we will be able to answer these questions. But make sure you at least revise the vitamins chapter for sure. Okay, we know about the vitamins. There are 2 kinds of vitamin, the fat soluble ones and the water soluble ones. Right? Fat soluble ones are the what? DAKE. D, vitamin D, vitamin A, vitamin K, and vitamin E. Right? So vitamin D, A, K, E. Right? And water soluble vitamins are vitamin B and vitamin C. Vitamin B and vitamin C. So how does B looks like? B looks like the number 8. Right? Why I'm asking you this? Because there are 8 B complex vitamins. There are 8 kind of vitamin B, right? So that's how you can remember it. That is vitamin B1, B2, you have B3. Do you have B4? No, you don't have B4, but you have B5. So there is no 4 in between. Do you have 6? Yes, B6 is there. Then B7 is there. Then B9 and B12. So there is no 8, no 10, and no 11 number. So you can remember it anyways. So these are the 8 vitamin Bs, and they have names associated with them. And then vitamin C is ascorbic acid. Vitamin C is also known as ascorbic acid. And what kind of disease it is related to? It is mostly related to the scurvy. Why scurvy? Because vitamin C is important for the hydroxylation of the proline and the lysine residues in the collagen synthesis. Right? If that hydroxylation, which enzymes are involved? This is another high yield topic. Lysyl hydroxylase, Lysyl, Lysyl, and prolyl hydroxylase. These 2 enzymes are responsible for the hydroxylation of the lysine and the prolyl residues during the collagen synthesis. And these enzymes, they both require vitamin C. If there is a deficiency of the vitamin C, then this hydroxylation does not take place properly. So the collagen is loosened. So that is leading to the scurvy. Scurvy is what? Scurvy is your bleeding gums. Bleeding gums. Bleeding gums. So if you have a question in the exam related to this, you know vitamin C, why the gums bleed? Because of the improper hydroxylation of the collagen, right? And another important aspect about vitamin C is, what is? It is an antioxidant. This is an antioxidant. And what kind of antioxidant? This is a water soluble antioxidant. There is a fat soluble vitamin also, which is an antioxidant. Fat soluble vitamin is also an antioxidant. What is that vitamin? Vitamin D, vitamin A, vitamin K, or vitamin E? Which one? कौन सा है? You should be knowing that. This is one of the previously asked question. right? In the meantime, think about it. When we will come back to the question, you can answer me that. Okay. What else vitamin C is? Vitamin C helps in iron absorption. Vitamin C helps in the iron absorption. Another importantly asked question in the exam. So this was about vitamin C. We are not going in detail beyond that. Just know this much for this session. Now vitamin B complex. Let's see vitamin Bs. There are 8 kind of vitamin Bs. If you pick first 4, means vitamin B1, B2, B3 and B5, right? They are energy. They are energy releasing vitamins, because they are used by the enzymes. The cofactor form of these vitamins are used by the enzymes. They participate in the processes or the pathways such as the glycolysis, fatty acid oxidation. Those pathways are energy releasing, right? So enzymes involved in the energy releasing pathways require these 4 kind of vitamins. Hence they are also known as energy releasing vitamins. Okay. Now there is this B9 and B12, the last 2. So first 4 and the last 2, like these 2 are your hematopoietic. These are related to the synthesis of the iron. Hematopoietic vitamin. And B6, which is your PLP, is another very important vitamin. Okay, so you should be knowing major things about this. So you know there is a water soluble vitamin, fat soluble vitamin. Water soluble is B complex. 8 kinds are there. First 4 are your energy releasing. Last 2 are your hematopoietic vitamins. Vitamin C is ascorbic acid, antioxidant, helps in absorption of the iron, right? They are interrelated to each other. It is related to the scurvy because of the hydroxylation of the lysine and the proline residue during the collagen synthesis, right? Now about the fat soluble vitamin, vitamin D, vitamin A, vitamin K, vitamin E, right? You should be knowing all of them. So I'm going over few of the vitamins. So first let's talk about vitamin B1 in detail, little bit in detail, because there can be a clinical case scenario that can be given to you in the exam. So that is vitamin B1, right? Vitamin B is the first vitamin of the vitamin B complex. This is also known as thiamine. This is also known as thiamine. They can ask you if the levels of concentration of thiamine goes down, right? How do we test it, right? Which enzymes helps in the testing, right? That is your TK, trans-ketolase enzyme. Trans-ketolase enzyme is measured for the to measure the concentration of the thiamine. What decreases the thiamine level? Alcohol. Like those who are chronic alcohol, they are drinking every day. The alcohol reduces the absorption of the thiamine, right? And there are diseases that are associated with the thiamine. Most of the times we talk about beriberi diseases. Beriberi. Okay? Beriberi is a thiamine deficiency disease. There are 3 kinds of the beriberis. What are those kind? First one is the dry beriberi, then wet beriberi, and then infantile beriberi. These 3 are what? These 3 are your beriberis. And then there are neurological problems associated with the thiamine also. Those include, sometimes they just say it the Wernicke-Korsakoff syndrome. Just the Wernicke-Korsakoff syndrome. Wernicke-Korsakoff syndrome, right? Which is composition, which has like the patient will be presented with the Korsakoff syndrome issues and also the Wernicke encephalopathy, right? What happens in the dry beriberi? You should be able to figure it out in the exam. In the dry beriberi, patient shows polyneuropathy and the symmetrical muscle wasting. Whereas in the wet beriberi, it is related to the heart. So high output cardiac failure is the major symptom shown by the patient. Infantile beriberi. Infantile beriberi are associated with the newborn babies who are still breastfeeding and the mother was having beriberi during the pregnancy and after the pregnancy also. At that time, those babies, those infants, they have the infantile beriberi. If supplemented with the thiamine supplements, it can be cured. Wernicke encephalopathy. Wernicke encephalopathy is acute, it is reversible, but it is life-threatening neurological condition. Right? What kind of neurological conditions? It is CONA, not coma, but CONA. Confusion, ophthalmoplegia, nystagmus, and the ataxia. Korsakoff syndrome, patient shows in the Korsakoff syndrome, patient shows the amnestic disorder. Why? Because of the continuous intake of excessive alcohol, right? Confabulation. They will come up with something, the made up stories which really never happened. Personality changes and the memory loss. If we combine both the Wernicke encephalopathy and the Korsakoff syndromes together, that gives the Wernicke-Korsakoff syndrome, which is a higher state of the thiamine deficiency. When the patient is experiencing the thiamine deficiency over the prolonged period of a time, then they show the Wernicke-Korsakoff syndromes, right? Both the neurological as well as like all these confabulations also, and the eye-related ophthalmologia, nystagmus, everything will be shown by the patient. Okay, so this is the vitamin B1, right? Vitamin B1 is, how do we measure the vitamin B1 in the body? By the TK. So it is mainly because of the chronic alcoholism, right? जो बहुत ज़्यादा पीते हैं, those who are heavy drinkers, right? So for that you can remember those people who are doing the PK, they are drinking the alcohol. Drinking the alcohol means they are PK. So we measure their TK, right? This is the way you can remember it. Now let's move to vitamin B2. What is vitamin B2? Vitamin B2 is also known as riboflavin. What is the coenzyme form? Coenzyme form of vitamin B2 is your FMN, FAD. So you can remember this, that this is 2. So there are 2 coenzyme form, right? And how do we measure it? Via the succinate dehydrogenase. And the symptoms are chiliosis, magenta, colored tongue, and the corneal vascularization. Another importantly asked question, like in the previous recall questions, this was asked. Vitamin B3, okay? Vitamin B3 is your niacin, right? And how do we synthesize the niacin? Can we have the niacin in via the food or the animal products? No. Niacin is synthesized in the body from the amino acid tryptophan. So this is an atypical vitamin. Vitamin K is also a atypical vitamin. So vitamin B3, vitamin K, right? And also the biotin. These are atypical vitamin in the body, right? Niacin is synthesized from by the bacteria flora also, and also from the tryptophan we can synthesize it. And if the tryptophan deficiency is there, the patient will be experiencing what kind of a pellagra symptoms that we will discuss. Where do we see the niacin? It participates in the all kind of redox reaction. It is associated with the redox reaction, means the dehydrogenases, right? dehydrogenases. Dehydrogenases are the enzymes when the oxidation reduction has to take place, right? And it is synthesized from the tryptophan. Is this vitamin is related, synthesis of this vitamin is related to the other vitamins? Yes. Vitamin B2 deficiency and B6 deficiency also affects the synthesis of the niacin, right? And it used, vitamin B3 is used in treating dyslipidemia. Dyslipidemia is what? If you have increased HDL level and decreased VLDL level. That is dyslipidemia, and vitamin and niacin is given for the treatment, right? And what are the deficiency symptoms? Glossitis is the one, and the bigger major deficiency system we talk about is the pellagra. Pellagra is what? It is also known as D3. D3, your 3D. What these D's are? So you can remember this way. B3 is giving rise to D3, and this D3 is dementia, dermatitis, and diarrhea. And if it is not treated, it may lead to death of the patient, right? In the conditions when the patient is suffering from the heart nerve disorder or the carcinoid syndrome, right? These kind of patients, they also show the pellagra symptoms, right? And what else is seen in the niacin? The Cazal's necklace. That around the neck, a patch will be seen, kind of a necklace, right? That is the Cazal's necklace, right? Even the heart nerve disorder and the carcinoid syndrome is related to this, because this is related to the niacin deficiency. What happens in the carcinoid syndrome? We have discussed this before also in various other videos, but let's discuss it again. Okay, heart nerve syndrome is autosomal recessive, and that is because of the TRP deficiency. Why the tryptophan is deficient? Because of the deficiency of the transporter. Transporter is not there to transport it, right? So there is a decreased absorption from the gut. Decreased absorption from the gut. That is leading to the pellagra-like condition. That is heart nerve disorder. In the carcinoid syndrome, that is a cancer kind of a syndrome. What is going on is the TRP, as you all know, has two ways. Either one pathway is which leads to the niacin synthesis, and one pathway is leading to the serotonin synthesis. Right? In the carcinoid syndrome, most of the tryptophan is routed towards the serotonin synthesis. That leads to the decreased production of the niacin, leading to the pellagra-like condition. So based upon what examiner is asking you, they may give you just the pellagra-like condition. You should be able to figure out whether this is a heart nerve disorder or the carcinoid syndrome, or just a pure pellagra symptoms are there, right? Excess is also bad in the niacin. It leads to the facial flushing and hyperglycemia, and the hyperuricemia is for the excess of the vitamin B3 consumption. Next is vitamin B5, right? B5. 5 is pento, right? So the name of this is pantothenic acid or pantothenate, right? 5 here and 5 here. For 5, and you have to remember it is related to foot syndrome, Burnick foot syndrome. That is one of the previously asked question also, right? They may ask you this time in one or the other way. So remember this. 5 is pento. The name is pantothenic acid. Okay, so this is about the pantothenic acid, right? And it is a component of, if they ask you the question which enzymes it is related to, it is a component of the coenzyme A. Right? Coenzyme itself is a cofactor for the acyl transferases. So we see it near the fatty acid synthesis also, right? And the deficiency involves dermatitis, enteritis, alopecia, and the adrenal insufficiency, which is leading to the burning foot syndrome. So you have to memorize this. The burning foot syndrome is related to P5, that is pantothenate. Vitamin B6, very, very important enzyme, very. So many pathways, so many enzymes, they need the vitamin B6. What is vitamin B6? Vitamin B6 coenzyme form is PLP, right? We know about that. That is PLP. PLP stands for pyridoxal phosphate. That is a coenzyme form. Yes, you have to know all the coenzyme forms for the vitamins, because usually vitamins are not as such used by the enzymes in the form of a cofactor of coenzyme. They have to be converted in this form before, right? And where do we see the vitamin B6? We see in the various transamination reaction. Majorly, where do we see it? With the ALT and the AST also, right? In the urea, in the amino acid degradation, we have seen that before the urea cycle. It is associated with the decarboxylation reactions also. with the glycogen phosphorylase. Where do you see the glycogen phosphorylase? In the glycogen metabolism. During the fasting state conditions, the liver glycogen is broken down, right? That is the enzyme. There is the glycogen phosphorylase, which is a rate limiting enzyme, another very high yield topic for the exam. It is related to the, it is required for the synthesis of the various things, that is glutathione synthesis, cystathione synthesis, heme synthesis, niacin, histamine, and various neurotransmitters such as serotonin, epinephrine, norepinephrine, dopa, GABA, right? They all require vitamin B6. And the deficiency of the vitamin B6 includes convulsions, hyperirritability, right? And the peripheral neuropathy. Peripheral neuropathy is usually induced by what? The isoniazid therapy. So another high yield topic here, right? Those who are on the isoniazid therapy, what kind of vitamin deficiency they will be facing? Vitamin B6 deficiency. Even people who are taking for the long term the oral contraceptives, okay? It also leads to the sideroblastic anemia, the deficiency of the vitamin B6, right? So you should be knowing about all these things about the vitamin B6. PLP, we see in the transamination reactions. It participates with the decarboxylase, and the glycogen phosphorylase requires vitamin B6, right? Now, vitamin B7, that is another vitamin I want to talk about, because there are like two important topics related to the important questions that might come from the biotin. That is vitamin B7. How do I remember the B7 name? T is similar looking as the 7. So just know it, this B7 is the biotin. And it is a cofactor for the carboxylation reaction. Yes, B6 was also, but this one is the major cofactor for the carboxylation reaction. What kind of carboxylase? PC. PC stand for pyruvate carboxylase. Pyruvate carboxylase helps in conversion of the pyruvate to oxaloacetate. Oxaloacetate, right? Pyruvate is a 3-carbon compound. Oxaloacetate is a 4-carbon compound, right? So one carbon is like fixed into pyruvate via the carboxylase enzyme. Then ACC. ACC is acetyl coenzyme A carboxylase. It also requires biotin as a cofactor. PCC. PCC is propionyl coenzyme A carboxylase. Propionyl coenzyme A carboxylase, acetyl coenzyme A carboxylase. So PCACC and again PCC, right? And the deficiency is relatively rare, right? So one thing you have to memorize this thing, it is related to the carboxylation reactions. These carboxylase is requires the vitamin B6. Another important thing you have to remember is for the prolonged consumption of the raw eggs. Those who are eating the raw eggs, for the prolonged period of the time, right, they have the biotin deficiency. What happens is in the raw eggs, there is a protein that is avidin. Avidin protein is found in the raw eggs. This avidin proteins interferes with the biotin absorptions, which leads to the biotin deficiency in the body, and then all the carboxylation things are not taking place, and it is life-threatening. It is life-threatening, right? Though the deficiency is relatively rare, but prolonged consumption of the raw eggs maybe more than a week, 10 days or a month leads to the biotin deficiency, and you, like person who is consuming it, definitely is gonna get hospitalized. Right, so these are the few things I wanted to talk about. Few of the vitamins, yes, there is vitamin B9, B12, vitamin A, D, E, K, all those, right? But because due to the shortage of the time, I'm not going to go over every single one. Question number 1 was there. First question was fat soluble vitamin with the antioxidant effects. fat soluble vitamin with the antioxidant effect. Which fat soluble vitamin? We have four fat soluble vitamins D, A, K, E, right? Which is the having the antioxidant effect? It is vitamin K. Is it vitamin K? No. Remember, antioxidant, this one. So vitamin A is antioxidant. So don't forget with the K and A. Most of the students, they make this mistake. Vitamin A starts with the antioxidant. So remember, this is the fat soluble vitamin. Which water soluble vitamin is the antioxidant effect? That is vitamin C. That is vitamin C. Which vitamin deficiency leads to hyperemesis gravidarum? Right? In the pregnant ladies, you have this morning sickness. Why that is happening? Because of the deficiency of which vitamin? Because of the deficiency of vitamin B6. Vitamin B6 deficiency, right? Which vitamin enhances the iron absorption? Vitamin C is related to the absorption of the iron. Vitamin K2 is known as, vitamin K is a fat soluble vitamin, and there are three forms of vitamin K. K stands for coagulation, so it is related to the clotting factors. Clotting factors 2, 7, 9, right? 2, 7, 9. Clotting factor 2, 7, 9 and 10, and also protein C and protein S. And there are three forms, K1, K2, K3. These three forms of the vitamin K is just basically from which source they are coming from. Vitamin K1 is K1 we get from the plant source. So it is known as phyloquinone. Right? K2 is an like the animal source, you can say, not the plant source. So it is menaquinone. Menaquinone. K3 is the synthetic vitamin that we have synthesized in the lab by the human beings. That is menadione. So question is, which is the K2? K2 is your menaquinone. Okay. Biotin acts as a cofactor coenzyme for which kind of enzymes? If they give you the name of the enzymes, we know three enzymes it works with, right? That is pyruvate carboxylase, or what else? Acetyl coenzyme A carboxylase and propionyl coenzyme A carboxylase. These three enzymes it works with. Otherwise, they may give you the option with the carboxylation reaction. Next question is, which vitamin deficiency causes what? Burning foot syndrome. Right? For foot is 5. So we are talking about vitamin B5, that is pantothenic acid. Okay. Which enzyme level is tested in the thiamine deficiency? Usually thiamine deficiency is what we talk about for the chronic alcoholics. So people, they after PK, we check their TK, right? PK is we are calling a name of a person कि उसने बहुत सारा शराब पिया है, right? They have consumed so much of the alcohol. So PK, people को हम लोग trans ketolase का level we check it for, right? So this is a few things, very important few things about the vitamins. Let's move forward. There was this question, previously asked question, similar to this kind of a question, that is related to what? This is related to the electron transport chain, right? The question is saying that patient is undergoing an appendectomy under the general anesthesia, such as succinylcholine or inhaled anesthetics. When she began to develop muscle rigidity, tachycardia, and hyperthermia, which of the following best describes the mechanism of this process? Uncoupling of the electron transport or phosphorylation, inhibition of the NADH dehydrogenase, inhibition of cytochrome oxidase, inhibition of adequate supply of ADP. Right? We all know by this time that the anesthetics, they interfere with the electron transport chain, and most of them are what? They are uncouplers. They are uncouplers. And they uncouple what? The name, what electron transport chain is doing? It is doing the oxidative phosphorylation. Know the essence, ठीक है, कि what electron transport is supposed to do. Electron transport chain से what we are getting ATP. ठीक है, this is the major task of the electron transport chain. And this ATP, for this ATP, what we are doing? We are carrying out the phosphorylation of the ADP, right? ADP gets phosphorylated to give you ATP. So that is one aspect, that is phosphorylation, because the energy needed to convert the diphosphate into triphosphate is coming from its sources. Electron transport chain का resources क्या हैं? That is NADH and the FADH2. Right? What they give is they give the electrons. Right? Electrons से क्या हो गया? Oxidation. Right? So because of this electron transport chain thing, that is the chain is going on to carry out the transport of the electrons, right? That leads to the phosphorylation of the adenine diphosphate to the adenine triphosphate. So combining two of them together is oxidative phosphorylation. If this is falling apart, okay, if this is separated, those are known as uncouplers, right? Oxidation and phosphorylation, oxidation, phi phosphorylation, they are couples. If we break the couple, separate the couple, those are uncouplers. Okay, so most of the inhaled anesthetics, they are oxidative, they are uncouplers, we should rather say. They uncouples the oxidation and phosphorylation, right? So the answer to this is what? Uncoupling of the electron transport and the phosphorylation. So electron transport is your oxidation half and phosphorylation is your last half. If you can remember the electron transport chain, there are 5 complexes we talk about in electron transport chain, right? Though it is 4 are shown here, but there are 5 of them. The last one is ATP synthase. This is carrying out the phosphorylation. And there is, this complex is 1, this is 3, this is 4. What it is not shown here is complex 2. Complex 2 is not shown here, right? Complex 2 gets electrons from FADH2. Complex 1 gets electron from NADH, right? This transports, 1 transports its electrons to the 3 and the 2 also transports its electrons to the 3 itself, right? So 1 and 2, they don't see each other. Then the third complex can transport the electrons to the 4, right? And then finally what happens is when the electron is being transported from complex 1 to 3 to 4, every time there is, while the electron transport is carried out, there is a export of protons in the in between the outer mitochondrial membrane and the inner mitochondrial membrane. So if this is outer mitochondrial membrane, if I'm saying this is inner mitochondrial membrane, there is this inner mitochondrial membrane space. Okay? This is what? This is inner mitochondrial membrane space. So every time electron, when it is being transferred from complex 1 to 3 to 4, right, the protons are pushed outside in this mitochondrial, like this space between the 2 mitochondrial membranes. These protons are supposed to go via ATP synthase towards the inside of the cytosol, right? And that is what? That is kind of a turbine. If you see it, protons are going to push, the turbine is going to get rotated. This turbine is ATP synthase. That leads to the synthesis of the ATP. This is bringing out the phosphorylation, right? And that what it is known as a chemi-osmotic theory also, right? Because if there is lots of positives here, right? Outside, there will be negative. That is making an electrochemical gradient. Wo energy is too much. So in one uncoupler, what happens is this is being disturbed. Why? The protons, ATP synthesis, they are not going to come in, but they will be rerouted. Wo kisi se bhi, they will come inside. Now don't say ki they become, yeah, you can say they have become more permeable to the inner mitochondrial membrane. So all the protons, they will come inside without going via the ATP synthase, which was a standard way to come inside. So protons are being lost. Protons are being lost, but because of this gradient that was happening, what is there? There is a loss of energy. This energy was supposed to be used for the generation of ATP, लेकिन वो नहीं हुआ है. That is why that will lead to increased body temperature. Why body temperature will be increased? Because this will be lost as heat. That is why the patient is experiencing hyperthermia. देखो, the patient is experiencing hyperthermia because जितना भी proton है is coming inside as such, not via the ATP synthase. Okay, so this is the what uncouplers does. So in uncouplers, you will see the rise in body temperature or the fever. This term you will see. Okay, now you should be knowing about the other inhibitors also. So what you have here is you have complex 1. They are not going to give you as like very simple question in the exam ki complex 1 is there, complex 2 is there, complex 3 is there. Complex 1, complex 2, complex 3, complex 4, and then finally is the complex 5, which is known as ATP synthase. ठीक है, वो ऐसे नहीं देंगे because that is going to make your life very easy. You have to know the name of the complexes. Complex 1 is getting the electron from what? NADH. So the name is NADH dehydrogenase. Complex 2 is getting the electrons from FADH2. FADH2, if you can recall, is produced in the TCA cycle also, right? And who was doing it? When you are synthesizing the succinate, right? So that there you see it. So remember, this is succinate dehydrogenase. This is succinate dehydrogenase, right? And complex 3 is cytochrome oxidase. Complex 4 is cytochrome reductase. Cytochrome reductase. Complex 5 is ATP synthase, or you can say F0F1 particle. Why? Because the way it is, the structure is there. So this is the F0 part and there is this F1 part, right? This is the world's smallest turbine. This is how your ATP synthase looks like. It has F0 and F1. F0 is just a passage so that protons can come in. When the protons are coming in, the F1 rotates. It looks like a fan which has 4 blades, and those blades are units, right? So when they accept the protons, that is carrying out the phosphorylation. So this part here, it rotates. It rotates. This is known as F1. F0, F1 particle. Okay, so these are the name of the complexes and you should be knowing about their inhibitors. How you are going to memorize it? Complex 1, right? Complex 1. So inhibitor name is rotenone or phenobarbital. Easy, right? This is easy. Succinate dehydrogenate is inhibited by malonate. Usually not asked too much. Cytochrome oxidase, that is complex 3, is inhibited by antimycin. Not so popular. What is popular is what they ask in the exam is the complex 4. Most beloved question of the examiner. Complex 4, that is cytochrome reductase. I'm sorry. My apologies. This is cytochrome reductase. This is cytochrome oxidase. My apologies, really my apologies. This is RO. So this is oxidase. Complex 4 is oxidase. Okay? Complex 4 is oxidase. So this is how I remember it is CO. That is how I got कि मैंने गलत लिखा है. That is CO. CO is carbon monoxide, which is a gas. So when I say gas, so I have told you earlier also, all the gases are going to inhibit the CO. CO, CN, hydrogen sulfide. These are all gases are going to inhibit the complex 4. Most commonly asked question. How do they ask the question? Either they will give you a scenario that there is a fire that is taking place, the fire people, they went in, right, and one of the person was admitted to the hospital, and this is going on. So probably if there is a fire is breaking out, we expect carbon monoxide to be there. Okay, carbon monoxide is there. So he must have inhaled carbon monoxide, which inhibited the complex 4, that is cytochrome oxidase, CO oxidase. My humble apologies for that. Now, 5 is what? ATP synthase, right? And those who are uncouplers, they inhibit this, right? So we have many kind of uncouplers. Majorly, they give you 2,4-DNP, 2,4-dinitrophenol, but there are physiological uncoupler also. There are physiological uncoupler. In the brown fat, there is thermogenin. Bears hote hain na, who go under hibernation, right? For the 6 months, they sleep for the 6 months. So during those periods, what they consume is they consume their fat, stored fat, as a source of energy. That brown fat we know called as, like, that contains thermogenin. That is physiological uncoupler. Who else? Thyroxine hormone is physiological uncoupler. Free fatty acids are physiological uncouplers. So just to give you few names, you must see a question out of this in the exam related to the electron transport chain. So make sure you remember it very carefully. Okay, let's move on to the next topic, okay, or next question. There is this 9-month-old child is presented to the emergency room by his parents, who report that he has been vomiting and had severe diarrhea. ठीक है, severe diarrhea है, vomiting है, and the episodes of vomiting begin when the parents started feeding their child as a cow milk. ठीक है, here is when they say it ki weaning off was going on, right? Baby was taken off from the mother's milk, but table thing is given. If they give you the table food, think about the fructose metabolism is going on. But they are telling you the cow's milk. Cow milk is lactose. So it means lactose kind of a deficiency is going on with the baby, that is resulting in the vomiting. Laboratory tests showed elevated blood galactose. ठीक है, that makes sense also. Galactosuria, metabolic acidosis, albuminuria, and hyperamino aciduria. These clinical and laboratory findings are most consistent with which of the disorders? It is, there is only one option related to the galactose, that is hereditary galactosemia. Why we are not picking the others? Alkaptonuria is because of the amino acid, right? Alka shouted, oh, ochronosis. Urine turns black upon standing, right? That we have discussed in the previous video. That is alkaptonuria, like the black pigments on the pinna, right? On the nose also, in the eyes also. That is alkaptonuria. Essential fructose urea is not problematic too much, but definitely if the fructokinase enzyme is absent, that is leading to the essential fructose urea, and that is usually like benign condition. It gets detected when the urine testing is going on, only then they are going to find it out. The Menkes disease is related to the copper. What other disease is related to the copper? That is the Wilson's disease. So you should be able to figure the difference out. Okay, let's talk about the galactose here now. One of the favorite question from the galactose. Know that the glucose and the galactose, they are C4 epimers. What do I mean by C4 epimers? That this OH, just this position of this OH, which is at carbon number 4, is different. Rest everything remains the same. So if you just rotate the OH to 180 degrees, then you can convert glucose into galactose and galactose into glucose, right? So that is what is the another question related to this part, galactose part. They are C4 epimers, know that. Now let's move on to the question. Question was telling you about the galactose problem. What happens is, in the cow's milk, we have lactose. Cow's milk contains the lactose. Any kind of a milk contains the lactose. Lactose is what? Lactose is a disaccharide, right? Lactose is a disaccharide. So it is broken down by enzyme Which enzyme? Lactase. Lactase breaks it down and that gives you galactose plus the glucose. Right? Glucose can be used by the body as such, but the galactose digestion is a problem. Yes, you can say ki ma'am, let's take epimerase enzyme, or since they are C4 epimerase, it will change them. No, there is no galactose epimerase as such present. Right? Seedha route nahi hai. You have to take an alternative route to convert the galactose into glucose. How that is done? This is done like this, which we have studied under galactose metabolism. Okay? So when the galactose is ingested, right, galactose is ingested in the body, it comes inside. So you can imagine galactose was outside, it came inside, where? Inside the cell where we want to digest it. So since the cell membrane usually allows the free movement of galactose in and out of the cell, first we need to phosphorylate it so that it gets trapped inside the cell so that it can be digested. Same kind of phosphorylation we do when, remember, glycolysis mein first step is glucose ko we are converted into glucose 1-phosphate by the kinase enzyme. So glucose is trapped inside the cell so that process can be taken place. Same thing is the fructose also. Fructose is converted into fructose 1-phosphate by the fructokinase enzyme. So it is trapped inside, it cannot go outside, and then we carry out the metabolism. Galactose is also converted into galactose 1-phosphate. Enzyme is kinase. What kind of a kinase? Specific to galactose, so we are calling it as galactokinase. Yes, it can be hexokinase, right? Hexokinase is a non-specific kinase. Hexo means any kind of a hexose sugar can be converted by hexokinase to its phosphorylated form. Then there is uridyl transferase. Okay, this is the part where the galactose is converted into glucose. See here. I'm gonna erase this. So see, there was galactose here. There was galactose here. Now this galactose becomes what? Glucose. And who is doing it? Uridyl transferase is doing it. That means we are talking about the involvement of what? Uridine. How uridine is getting involved here? This is UDP glucose is an entity. The UDP, you can remember it as like all of you have been to wedding. When the groom comes, right? Groom comes on the chariot. So your groom, the major person is your glucose. He has to sit on a chariot and the chariot take it to the wedding place. So imagine like that. Okay? So this is your chariot. Okay? My drawing is not good, and this is going to carry your glucose so that we can convert the galactose into glucose, right? So this is what? This chariot is your UDP. UDP. It will take it to the venue, right? And then the galactose will be exchanged for the glucose. And then the UDP is going to leave with galactose, right? So when it will be leaving, the chariot will not have the groom, that is glucose, rather it will be having galactose, right? So now this UDP galactose, is converted into glucose by the epimerase. Okay? It is like a main process was to convert the galactose into glucose. This part, entire part is a main pathway. So we don't make changes over there. We just take the galactose out from that main pathway and then the epimerase converts that galactose into glucose. Now, let me recite it or revise it again so that it makes sense to you. So, galactose is coming after the digestion of the lactose by the lactase enzyme. Okay? So, in order to trap this galactose inside the cell so that metabolism can be done, the kinase phosphorylates it by using 1 ATP and galactose becomes galactose 1-phosphate. Then there is transferase enzyme, uridyl transferase enzyme, which switches the glucose for the galactose so that galactose 1-phosphate becomes glucose 1-phosphate. Okay? This glucose 1-phosphate can be used, can go either glycolysis or the gluconeogenesis pathway. We know about that. Now the question remains, this UDP glucose becomes UDP galactose, right? And this UDP galactose is converted into UDP glucose by the epimerase enzymes. Why? What kind of epimerase? 4 epimerase. So remember this, since the glucose and the galactose are C4 epimers, so the name of the enzyme is epimerase, right? Now this enzyme is, if this enzyme is deficient, also known as GALT enzyme, that leads to galactosemia, right? So galactose metabolism disorder is either you have the galactokinase which is deficient or the GALT deficiency, right? GALT deficiency leads to the classical galactosemia that is shown in this patient, the baby. Now, this is one thing. They may give you that there is I is having a disorder. What kind of a disorder? Because of the accumulation of the galactitol. Since if this is not present, this will be remaining. This is going to inhibit galactokinase. So in the lens of the eye, right, there will be an accumulation of the galactose. In the retina, we know there is an enzyme aldol reductase, which is going to reduce this aldehyde. Galactose is an aldehyde. It is going to reduce this aldehyde into alcohol. And that alcohol is galactitol. And this galactitol gives rise to oil drop cataract, right? So those people who are suffering from the galactosemia, that is classical galactosemia, non-classical galactosemia, and the classical galactosemia is, there are two different versions, right? So if the aldol reductase is deficient, do you expect to see the cataract, the oil drop cataract? No oil drop cataract. Because there is no reductase to convert galactose into galactitol, which is giving rise to the oil drop cataract. Only in the classical galactosemia, you will see the oil drop cataract. And that classical galactosemia is because of the GALT deficiency. So in the galactose deficiency disorders clinical presentations, if they tell you specifically the oil drop cataract, then you should always be picking GALT enzyme, and the deficiency disease is called as classical galactosemia, right? Aldol reductase deficiency gives rise to the non-classical galactosemia, right? So I guess I have pretty much covered 4 or 5 questions that may come from the galactose metabolism, right? I don't think isse zyada koi bhi question banta hai from the galactose metabolism, okay? Now, which enzyme is capable of digesting cells and the cellular material? So we know there are several organelles inside the cell, right? We have the mitochondria, we have the lysosomes, we have the Golgi apparatus, the rough endoplasmic reticulum, the smooth endoplasmic reticulum. So many kind of organelles are there, and all organelles, they have a specific function associated with them. So where the digestion takes place? Digestion takes place in the lysosomes. There are these lysosomal enzymes which are responsible. Now, what if the lysosome enzymes are not available? Imagine a scenario, the gene is defective, right? Or let's take another real case scenario, right? There is this lysosomal enzyme, right? Where we are synthesizing it? We are synthesizing it in the cytoplasm, right? After the protein modification, like up to that part, let's talk about, okay? Lysosomal enzyme ban gaya hai, but still the digestion is not taking place, right? It is not recycling the material. That is going to lead to a disease known as I-cell disorder. So lysosomes will be showing what? Inclusion bodies. Because of the deficiency of the lysosomal enzymes. Why that is happening? When the enzyme, all enzymes are protein, okay? So they will be synthesized into the regular way. but the lysosomal enzymes will be specifically labeled with the mannose phosphate. Okay? If they have this kind of a flag, the enzyme is containing the mannose flag on them, they have the mannose tagged upon them, then they will be taken to the lysosomes so that they can perform their functions. But in many cases, they are not tagged by the mannose, so that leads to the deficiency of the lysosomal enzymes or non-availability of the lysosomal enzymes inside the lysosomes, giving rise to I-cell disorders, or it will form, because whatever the material that need to be recycled will be staying in the lysosomes. ठीक है, उनका degradation तो होगा ही नहीं because the enzyme is not available. That will lead to the formation of the inclusion bodies inside the lysosomes. Okay. What mitochondria does? Who can tell me what mitochondria does? It is an powerhouse of a cell which generates like lots of ATPs. Maximum ATP we are getting from the electron transport chain, TCA cycle, beta oxidation. Okay? All these are what the ATP releasing unit pathways. So all these pathways are taking place in the mitochondria. Yes, for examination point of view, you have to know in which organelle the pathways are taking place, right? The rate limiting step in glycolysis occurs at the step catalyzed by which of the following enzyme? Rate limiting step in glycolysis. In glycolysis, we just talk about 3 irreversible steps. Almost 10 steps are there. 3 irreversible steps are there. Rest all are reversibles. Out of those 3, we have 2 enzymes which are rate limiting enzymes, we call it. Majorly actually 1 rate limiting enzymes are called, but 3 irreversible steps, and 1 major rate limiting enzyme of the glycolysis is PFK. PFK, that is PFK1, phosphofructokinase or 6-phosphofructokinase 1. None other than that, okay? So let's talk about in detail about the glycolysis also. There can be many questions which can come from the glycolysis apart from this question. Glycolysis is what? Glycolysis is conversion of glucose into the pyruvate. When this conversion of glucose into the pyruvate will take place? Fed state or the fasting state? Usually it happens in the fed state, okay? And excess of glucose will be stored in the form of the glycogen as well. Okay? Excess will be stored in the glycogen, and some of the glucose will be undergoing via the glycolysis pathway to give you what? ATP. ATP to is needed, na? Right? And glucose is converted into what? Glucose 6-phosphate. This is carried out by 2 enzymes, glucokinase and hexokinase. Glucokinase is specific for the glucose. So when during the fed state, glucose concentration is very, very high. It has high Km. Glucokinase has high Km, right? So it has more, so whenever the concentration is very high, did I say it right? Glucokinase is having just affinity for the glucose. It is having affinity for the glucose when the glucose is at high concentration, which is usually in the fed state, then glucokinase work. Hexokinase is kind of a routine kinase. Any kind of hexase it is available, right? It can work with it. So this is converting glucose into glucose 6-phosphate, and this is a irreversible step. This traps the glucose inside the cell, and we know there are various kind of GLUT transporters, GLUT1, GLUT2, GLUT3, GLUT4, right? And GLUT4 transporter is the only one which is insulin sensitive transporter. Only the fed state this GLUT4 is working. Okay. Now glucose 6-phosphate is being formed, and this step is energy requiring step, means 1 ATP is being consumed in this step. Then glucose 6-phosphate is converted into fructose 6-phosphate by the isomerase enzyme. Glucose is an aldehyde, fructose is an ketone. So that is isomerization only. Right? The phosphofructokinase PFK1, which again utilizes 1 ATP. Right? This is PFK1. This is another irreversible step, and this is the rate limiting enzyme. Okay? If you are adding 1 ATP, it means you are doing the phosphorylation. So fructose 6-phosphate becomes fructose 1,6-bisphosphate, right? So there is the 6 carbon molecule At this half, you have one phosphate. At this half, you have the one phosphate, and you cut it off into two pieces. So you get three piece molecule. This also has a phosphate. This also has a phosphate. One is dihydroxyacid DHAP, and another is glyceraldehyde 3-phosphate. Right? Glyceraldehyde 3-phosphate is taken further down, and that is converted finally into what? The pyruvate. Right? And one important thing you should be knowing about here is, one more step is pyruvate kinase. Phosphoenol pyruvate, PEP, phosphoenol pyruvate to pyruvate is done by enzyme pyruvate kinase. Right? And pyruvate kinase deficiency is the second most common deficiency that is found inside the human beings. Right? Second most common enzyme deficiency. And this is also a irreversible step. This is also a reversible step. Right? So in the gluconeogenesis, if we say, okay, gluconeogenesis mein what we will do? We will be doing the reverse of this. So we cannot just take kinase. It is done via the PC, that is pyruvate carboxylase enzyme, that is PC, and PEPCK. PEPCK. Right? These are another important asked question in the exam. So just know about it. So in total, if you look at the energetics of the glycolysis, you consume 2 ATP. 2 ATPs are being consumed. 2 ATPs are being consumed. Right? At first step is glucokinase use karta hai, another phosphofructokinase use karta hai. These are the two ones. And then what you release is, you release ATP, one ATP here, and one NADH, right, by the glyceraldehyde 3-phosphate. Since there were two glyceraldehyde 3-phosphate that were being formed, okay, this and this. So you do the times 2. One NADH is how much ATP? One NADH is 2.5 ATP. And then you have one ATP. Right? And then another is by pyruvate kinase. That is one more ATP. So whatever is there, that is times 2 is the amount of ATP you get. Okay? That is how much? 5, 6, 7, 8, 9. Total 9 ATPs are being produced. 2 are being consumed. So net production of the glycolysis is 7 ATP. And that is under aerobic condition. That is under aerobic condition. What happens in the anaerobic condition? In anaerobic condition, this pyruvate will be converted into what? Lactate. And that lactate will be consuming your 1 NADH and will be converted into NAD positive. ठीक है? So it means जो NADH यहाँ produce किया था, whatever NADH was given here, right? It is being consumed by here. ठीक है? So it means there is total loss of 5 ATPs in the anaerobic glycolysis. So you just say -5 इसका हम करेंगे तो you are left with what? 2 ATP. So in the anaerobic conditions, glycolysis gives you just 2 ATPs. Are we clear about the energetics also? Right? Next, another thing I wanted to give you this chart. Right? Those who never attended my session before, just know this chart. This is very important for you to know. This includes all the pathways for the carbohydrate metabolism. Right? So you should be knowing their location and also the rate limiting enzymes or the rate limiting steps for the carbohydrate metabolism. Okay, the glycolysis. What is the another name of the glycolysis? EMP, Embeden-Meyerhof pathway. It is taking place in the cytoplasm and the rate limiting enzyme is PFK1. Gluconeogenesis is the synthesis of the glucose, and usually that takes place in the fasting condition, right? Because we have to compensate for the blood glucose level. It is taking place in the cytoplasm itself. So you can, now this is another question. Glycolysis is also taking place in the cytoplasm. Gluconeogenesis is also taking place in the cytoplasm. So it will lead to a futile cycle. So what happens is the very last step. Last step is this one. Converting glucose to glucose 6-phosphate and glucose 6-phosphate to glucose. So glucose to glucose 6-phosphate is glycolysis. and glucose 6-phosphate to this is gluconeogenesis. This was done by this was done by the kinase enzyme, and here is what your phosphatase enzyme, glucose phosphatase enzyme, that is going to remove the phosphate group. This enzyme glucose 6-phosphatase is present everywhere in the endoplasmic reticulum. So glucose 6-phosphate during the fasting condition is transported inside the endoplasmic reticulum, and then there is this enzyme phosphatase which will make the glucose and will release the glucose in the blood for the use. Okay, that is how the glycolysis and gluconeogenesis is balanced, and this kind of regulation mechanism is known as compartmentalization, right? Because the enzyme is available in different compartment, different organelle, that is endoplasmic reticulum. Another very important asked question in the exam. TCA cycle. TCA cycle is taking place in mitochondria, right? Mitochondria is a powerhouse. So all the energy releasing thing are taking place in the TCA. Means lots of ATP are taking in the TCA, right? And the rate limiting enzyme is isocitrate dehydrogenase. Glycogenesis is taking place in the cytoplasm. Glycogenolysis is taking place in the cytoplasm. But the rate limiting enzymes are glycogen synthase and the glycogen phosphorylase, right? Similarly, you can ask me, since they are taking place in the cytoplasm, same way glycolysis and gluconeogenesis is regulated, this is being regulated. And who regulates it? Insulin. You should be knowing that insulin regulates it. Insulin and the glucagon. Glucagon levels are up in the fasting condition. That time insulin is down, right? So glucagon does what? Glucagon is there in the fasting condition, right? And in the fasting condition, we have to break down the glycogen. We have to do the glycogenolysis, right? So at that time, the glycogen phosphorylase is activated because of the dephosphorylation. If the glycogen phosphorylase has a phosphate, that is a deactivated form, right? So how do I have what trick I have told you is IP. Remember IP. Insulin enzymes, like in the fed state, when the insulin level is up, the enzyme is going to activate the phosphatase. Insulin activates the phosphatase that is going to remove the phosphate group, making the synthesis enzyme active. So glycogen synthase, if this is getting this was phosphorylated, it means is this synthesis going to take place? No. Because synthesis is taking place during the fed state. Fed state is insulin. Insulin is going to activate phosphatase. And what is the job of the phosphatase? To remove the phosphate. I guess that is making more sense now. So IP. Remember this. In case you get confused, even many times I get confused, I have learned this series. That keeps my mind on the track. Okay? Insulin activates the phosphatase. Phosphatase removes the phosphate. So means enzyme is getting dephosphorylated. So enzymes getting dephosphorylated in the fed state are participating in the synthesis. So based upon the fact, you can figure it out. If the glycogen synthase has no phosphate, that is the active form. That is the active form, right? And this will get phosphorylated. That is inactive form during the fed state conditions. So this is how we balance the synthesis and the breakdown process. HMP shunt. HMP shunt, when the excess glucose is rerouted in the HMP, that is taking place in the cytoplasm, right? And that is rate limiting enzyme is glucose 6-phosphate dehydrogenase. The most common enzyme deficiencies. What are the products of the HMP shunt? You get ribose 5-phosphate and NADPH. NADPH. You have to know the products of the HMP shunt, right? Question is, which of the following options given below explains non-competitive inhibition? So you have non-competitive and the competitive inhibition, right? And there are two factors we juggle around, Km and Vmax. Okay? One thing goes up, one thing goes down. Remember, competitive, K ma'am goes up. Competition mein Michaelis constant is high, is up, right? So that is going to solve your half of the problem. What do we know? Michaelis-Menten equation, right? Michaelis-Menten is, this is the substrate concentration on the x-axis, and the Vmax. Vmax means, I should saying the velocity, the rate of the reaction on the y-axis. So this is the substrate concentration, this is the velocity, right? So as soon as you start adding the enzyme to the reaction mixture, enzyme will combine with the substrate, so the rate of the reaction is going to what? Is going to increase. Is going to increase when we are adding enzyme. Enzyme will bind to the substrate and that will give you the product. So more enzymes are added, the rate of the reaction will go up, 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 and after a particular point in time, no matter how many much enzyme you are adding, the reaction will reach to a saturation kind of a point. Right? There is no beyond increase in that, right? So that is the parabolic curve. That is a parabolic curve. And that is first order kinetics. Right? And this is the maximum velocity or maximum rate this reaction can achieve. It is different for different enzymes. Okay? This is the maximum for one thing. And this is the half of the Vmax. And if we drop down the half of the Vmax on the x-axis is the substrate concentration, we get Km. So Km is what? Km is the substrate concentration when the velocity is half of the Vmax. Okay? Adha Vmax is giving you the Km. Okay? Since this is a parabolic curve, getting an exact number is difficult, mathematically incorrect. So there is an advanced version, that is Line-Weaver-Burk plot. It is same, just know that is we are doing the inverse. Now, we are not just taking the velocity and the substrate concentration, we are taking the inverse of the velocity and the substrate concentration, and then we make a plot. In that plot, we get a line that extends beyond the zero. Left hand side pe jayegi, if you know, remember the maths, right? This is the negative, right? And this is how this is going to drop down. So y-axis ka same intercept is going to give you 1 over Vmax, and x-axis ka intercept is going to give you 1 over Km. Since this is towards the negative, it is minus 1 Km. They may ask you to label it, they may ask you to recognize this, okay? Now, in both the cases, whether we are talking about the Michaelis parabolic curve or the Line-Weaver-Burk plot, another question they can give you is, you have to recognize just by looking at the plot, whether it is a competitive inhibition, it is a reversible, irreversible, what kind of inhibition is that. So remember this one. The black is what? The black is the normal enzyme, right? That is what? This is a parabolic curve. Okay? This is a parabolic curve, and we know this will be a Vmax, kind of a Vmax. And this will be half. If we take a half and drop it down, that will be Km. Make sense? This is we have just discussed. In the competitive inhibition, if that is represented by the red color, that is represented by the red color. So, it is below, but it reaches to Vmax. So, Vmax is same. Vmax is same, right? Vmax is same, but Km is going to shift. If this is half of the Vmax, so, and if you drop it down, what do you see? Km has changed, or Km has increased. Increased Km ho gaya na? Earlier this was the Km. Earlier this was the Km. So there is this difference. Km is increased. That is competitive. Competitive mein Km increases. Good. Now, let's take another. In the non-competitive inhibition, non-competition may the reaction velocity will not reach to the maximum because it is inhibiting it. Non-competitive hai, binding to site other than the active site. So, this is kind of a graph you are getting. Okay? Towards the lower side. Do you see the Vmax? Vmax was this. Now, the Vmax has changed. Vmax has changed. That is decreased Vmax. That is decreased Vmax. But the Km in this case is the same. In uncompetitive inhibition, what happens is Km is increased and Vmax is decreased. Vmax is decreased and Km is increased. You have to know all these three conditions for the examination point of view. Now, sometimes they give you this plot. Just plot thingy and you have to figure it out by just looking at the Lineweaver bar plot, what kind of inhibition you are talking about. Okay? First is the straight line. Green is the straight line. Uninhibited enzyme. If it makes an X like this means this is what? This is your 1 minus 1 over Km. Km is increased क्योंकि वो 0 की तरफ जा रहा है. This is more negative. This is a bigger number. ठीक है? This is X. This is X. That is competitive inhibition. Non-competitive inhibition, they will make a V. Inhibitor will make a V. Uncompetitive, they will be very parallel to each other. ठीक है? But the inhibitor will be on the upper half, not on the lower half. So you should be able to recognize everything like this. ठीक है? So now the question asked you about what? The non-competitive inhibition. In non-competitive inhibition, what happened to the Vmax? Vmax lowers down, right? Vmax lowers down and the Km is Vmax lower down हुआ देखो. In the non-competitive inhibition, Vmax lowers down and the same Km. So you have the lower Vmax. Affect by both Vmax and Km? No, Km is same. Km is same. So this is not an option, this is not an option, this is not an option. This is the correct option. The disease Lesch-Nyhan syndrome occurs because of the following enzyme deficiency, and by this time, since you have exam in few days, that is HGPRTase enzyme, hypoxanthine guanine phosphoribosyl transferase enzyme, right? And that is related to what? That is related to your metabolism. What kind of metabolism? Purines degradation. Purines degradation. You know that purines, pure as gold, are adenine and the guanines, right? Small name, big structure, you should know when you have to do the de novo synthesis. De novo synthesis, right? You should be able to tell, they can ask you which atom is contributed by whom. We have discussed that in previous video that I have taken. I told you in the beginning of this session, so I'm not going to go over that again, but you should be revising that also. Okay, nitrogen, four nitrogens are there. From where these nitrogens are coming from where carbons are coming from. This is donated by glycine. These two nitrogens are donated by the glutamine. This nitrogen is donated by the aspartate. The topmost carbon is donated by carbon dioxide. These two carbons are donated by the tetrahydrofolate system. So we can either synthesize the purine like by the de novo synthesis or by the salvage pathway. In salvage pathway, when you are doing the degradation of AMP and the GMP, right? What you get in the end is hypoxanthine and the guanine. Okay? Guanine and hypoxanthine, they can be converted like using the salvage pathway. We are going about the salvage pathway. They can be converted back into IMP and GMP. So we are talking about just this pathway, right? Hypoxanthine and guanine can be converted into IMP and GMP by the enzyme HGPRTase, right? So in Lesch-Nyhan syndrome, this enzyme is absent, which is going to give rise to the Lesch-Nyhan syndrome symptoms. What are those symptoms? Spastic cerebral palsy, self-mutilation is the biggest thing you should be looking for. Hyperuricemia, early death, and this is what? X-linked disorder. Remember that. This is an X-linked disorder, right? So there is a deposition of the monosodium crystals in the joints, and that pains, they are needle-shaped crystals. There will be kidney stones that will be found, and the baby is gonna, like the toddler is gonna eat the lips, fingers, and we have to keep them tied up. Self-mutilation is there. Now, one student has asked me, right, in the purine and the pyrimidine metabolism, if there is an increased uric acid, right? There is an increased uric acid under some circumstances. Why it is leading to the increased, giving rise to the lactic acidosis? Or in another way, remember, the increased uric acid in the urine gives rise to increased lactic acid, gives rise to lactic acidosis. Why that is happening? Very, very important thing, right? I remember last time I was unable to answer this question, right? So when you have high uric acid in the urine, in the blood there will be increased lactic acid. Reason being, there is only one gateway for the excretion of both the uric acid and the lactic acid in your renal tubule, okay, in the renal tubule. So either the lactic acid can go out or the uric acid go out, right? So when the uric acid levels are high, uric acid is thrown out first. So that leads to the leads to the higher concentration of lactic acid in the serum. That is why the patient experiences lactic acidosis. Right? So know these facts about the Lesch-Nyhan syndrome also. It is X-linked, right? Mental retardation, spastic palsy. Now there is another disease, Kelly-Sieg-Miller syndrome, which is because of the partial deficiency. In these patients, they don't see the self-mutilation, only the gout is present, right? Now the last question is detection of ketone bodies in the urine. How do you detect the ketone bodies in the urine? Actually, there are two tests we talk about. One is the Rothera's test, right? The one is the Rothera's test. In Rothera's test, right, we add the Rothera reagent and what do we see? We see the purple colored ring. That is purple colored ring. The presence of the purple colored ring on the upper part of the urine sample is indicative of the ketone bodies, and ketone bodies are synthesized during the fasting state, and ketone bodies includes what? Your acetoacetate, beta hydroxy butyrate. These are the ketone bodies, right? And another is this ferric chloride test, in which you add the 10% of the ferric chloride, right? And that gives the Bodiax red color. That gives a very deep red color, right? And then you boil it. If after the boiling the color disappears, that is telling you the presence of the acetoacetic acid. So they may ask you how the acetoacetic acid is being tested. Now we are specifically looking for one ketone body, that is acetoacetic. So if after boiling the color disappears, it is acetoacetic. If the color persists, right, then there is a presence of the salicylates. There is a presence of the salicylate. This is to distinguish whether we are getting a false positive test or not, right? Rothera test may give you a test which we assume it's false, but during the examination we don't find any ketone bodies, then we should go for the second test, right? That will confirm us whether the color is because of the acetoacetate, which is a ketone body, or it is because of the salicylates. That is a confirmatory test for the ketone bodies, okay? So know about this also, and there are many more topics. Just please kindly review the previous videos. See, the lesson for today is, which even I am learning every day, nobody is perfect. Everyone makes a mistake, but some mistakes will teach you a great lessons and make you a better person, right? Neither you are perfect nor I am perfect, but we learn from mistakes and we correct our mistakes. That is the biggest thing we can do to improve ourselves. So I am improving myself and you do. And please, if you have any other further questions, just drop me a message. Everybody knows my Telegram ID. That is @DRVARSHAGUPTA, right? You can drop me a message and I will revert it back to you. Thank you and good luck. A very, very good luck for your exam.